So welcome everybody to our California Master Beekeeper webinar series. This evening we have a special guest from Oregon State University, Corvallis, Dr. Ramesh Sagili. He's an associate professor in the Department of Horticulture at Oregon State University, and he obtained his PhD in entomology from Texas A&M in 2007, specializing in honeybee research, right?
Yeah.
Yeah, go Texas. His primary research focus is honeybee health, nutrition, and pollination. Oh, Ramesh initiated the creation of the Oregon Master Beekeeper program, so he and Alina have a lot in common in that regard. And he chaired the Oregon Governor's Task Force on Pollinator Help. That's a big deal. His research program addresses both basic and applied questions to improve honeybee health and nutrition. And so the majority of his research projects are collaborative efforts involving stakeholders like beekeepers and growers and the like. So he's authored several important research and extension publications, and he's received several awards, including the esteemed Entomological
Entomological Society.
Society of America's Pacific Branch of Research Award, Eastern Apiculture Society's Outstanding Research Award, and OSU Outreach and Engagement Award. We're so pleased to have you here. We thank you for your generosity of time and support in dropping in and sharing your insight and knowledge with us this evening. Thank you, Ramesh. We would like to invite our guests here this evening, if you have a question throughout this webinar, to please put it in the chat, and we will have an open Q&A towards the end of our hour together. So I invite you, Ramesh, to share your screen, and we'll get this webinar going. Thank you.
Yeah, thank you, Wendy, and thank you all for spending some of your night time listening to this presentation. So let me see if I can share the screen here. Can you see the screen, Wendy? Okay.
Yes.
And it's full screen, right? Okay.
It is. Perfect. It looks great. Thank you.
Okay, perfect. So what I'll do is in the next, at least I'll spend 55 minutes and then leave some time for questions. Yeah, I always go over time. So yeah, so forgive me if I do that again. I know you're all enthusiastic to learn more, and I want to provide as much information as I can. So Varroa and nutrition management, I strongly believe that these are key to success of both bees and beekeepers. So that's the reason I chose, and it's very timely, right? You'll be managing your Varroa soon, and nutrition probably in the fall. So I think these are important topics. Again, I don't have time to go over a lot of different things between this Varroa and nutrition, but I'll try my best to provide you some glimpse of the research that we have done over the last few years, and also what is important for you as a beekeeper to understand some basics in terms of Varroa management and nutrition management. And if I have time, I have some slides I put at the last minute on impacts of wildfires on bees, but we'll see. If we have time, we'll go over very quickly at that topic as well. Okay. So before I start, I would like to acknowledge all these incredible lab members that have worked in my lab for the last few years, and some of them have already proceeded to different careers, but at least several of them are still in the lab. These include graduate students, postdocs, undergraduates as well. So let's start with Varroa, which probably you'll all agree with me how important this is. I know I don't want to rank any stressors on bees, but Varroa is probably one of the top ones in our list, and we need, and especially the very new beekeepers in the audience, you probably should be paying attention to Varroa management. this can probably sometimes make or break your beekeeping operation. So there are two different phases. We talk about reproductive phase and phoretic phase when we talk about varroa mites. So I'll not go in too much deep into biology here, but give you a little bit overview so that it's behind or at least in top of your mind when you're doing varroa management. So female enters the cell, right? So let me go to the next slide and show you what I mean by. So what you're seeing here is the phoretic phase for those new beekeepers. The mites are on the bees. They're feeding on their fat bodies and a little bit on hemolymph as well. Whereas this is the part that we usually don't pay much attention because you can't see the mites, right? These are in the reproductive phase. They're already hiding inside those cells and they are breeding there. The female mite has entered, she's laying eggs and the new progeny is being produced. So, but this is the easy one. Even visually you can't see mites because they're hiding between these sternites, and sometimes you may see, but just because either they're trying to latch onto another bee or they're trying to go into another cell to reproduce. Or maybe you are looking at your hive when your mite levels are too high. So that's why you are seeing a high probability of finding mites at that time visually. But usually they are pretty much good at hiding. So female mites, they will go into the cells that I showed you about 12 hours prior to the cell being capped, and they hide under the larval food, right? You see that white glistening food that is provided by the worker bees to the larva. So they hide there so that the mites can't see, the bees can't see them and pull them out. And once the larval food is consumed, the larva from the larva, the mites, they get out and they start feeding on the larva. So they'll make a small incision or a hole on the larva, and then they start feeding on the hemolymph and fat bodies from there. So the first egg is laid about 2.5 to 3 days after the cell is capped. And then subsequently, each egg is laid about 30 hours later. So about 5 to 6 eggs, I would say, on average, a female mite that gets into the cells will be able to lay. And then usually the first egg is the male, and anything that is laid after that every 30 hours would become a female. So there is this inbreeding going on, and I want you to think, I will not have time to discuss all that inbreeding problem here, but isn't it amazing? Like inbreeding, we all know is not good for any organism, right? But in this case, in mites, there's a lot of inbreeding going on. This is the male that comes out first or hatches first will breed with the subsequent sisters that are being produced. So think about that. There are some speculations on why this is not a problem. Inbreeding is not a problem in all organisms. So it's an exception here. So I talked about this. So if you didn't understand anything that I said, just watch this animation. So here you have a feral hive or a honeybee hive that you are managing. So it's the same thing. We have bees, there are mites. So a mite goes into this cell. And you see it went all the way down and it's hiding, and now the cell has been capped. So the mite comes out and starts laying eggs. So that probably was a male, and these all will become females. And see, as you know, mites are not insects, so we use different terms like deutonyms, protonyms for their young stages. We don't call them larva or pupa. So here you see a bee coming out of the cell, and you see a mother that is there successfully comes out with the daughter. So usually if it's a worker cell, you could expect one. So the female mite that went in, the mother will come out with a daughter. But if it's a drone cell, they can get two individuals. That means a mother comes out with two more individuals that will be her daughter. So again, the drone life cycle, it's 2 days more, right? Or 3 days more, right? So 21 days for worker, 24 days for drones. So the mites are looking for, that's the reason when you read in books or in your California Master Beekeeper Program literature, you might have read that the mites are looking for drone brood, because that's a reproductive success strategy, because they can produce more. That means they're getting 3 more days to mate with that male, right? Mating is the trick. They already have produced 5 or 6 of those individuals, but they all don't get mated because the time is of essence. So if you give them 3 more days, there'll be more females that can mate with that brother, and they'll be successful as individuals. If they're not mated, then they're not any good for the hive. So for the mite population. So that's it, the bar. And again, I'll not go in deep. You probably learned in your program the symptoms, but I'll just very briefly mention here so that you have a kind of a refresher on the symptoms. So we use the term parasitic mite syndrome because there's not just one symptoms, there are multiple symptoms associated with mite problem. So you can see the deformed wing virus here. You see that bee with a deformed wing. So these bees don't survive more than a day or two. They can't really fly. They have already high titers of virus in them. You see, and you see the spotty brood pattern you see in this image here. The pattern is very spotty, right? So some of those cells are empty because either the bee died and the developing bee died and the bees have removed those late pupal stages or the developing bee inside those cells. So you see a spotty brood pattern. Again, spotty brood pattern can be symptoms of other things as well, right? For brood diseases, if you have read about all those things. So you have to look for several things. So bees with deforming virus, there will be some larvae that are young, like C-shaped still. They appear to be melting, but with no odor, because with AFB or EFB, there will be some smell associated, but you don't see that much with this. And a lot of uncapped pupal cells, like you see here. So this is another blown up image of the same thing. You see some of these cells that have been opened. So there is a reason why they have opened is because these bees have probably detected mites inside those cells, and they're trying to uncap and remove the mites from those cells. That's why you see a lot of uncapped cells with the bees already almost developed. Maybe in 3-4 days they would have hatched. So this one, I want you to pay attention here where I have circled. You see some white stuff, right? And probably most of you know that's the mite frass or the feces of the mites. So you can see that white sprinkle like crystalline structure. And sometimes, if you're feeding sugar, sometimes there might be some sugar as well. Don't get confused with the sugar versus the mite feces. There is a difference. If you have seen enough of mite feces, you will not get confused. So it's usually stuck to the top of the cell here on the top edge. And this is another image. If you hold them, you have to hold in an angle. If you're holding a frame straight, probably it's hard for you to see those. But if you have a good sunlight and if you're tilting and looking at an angle, you can see here, this is all mite frass or mite feces. If you see those, again, this is kind of an autopsy you're doing after the colony has died. So again, I'm showing you some colonies that have already died with mite infestation. So this frame is from one of those colonies. So you can see a lot. And sometimes I know it's unfortunate that you may lose a hive, and I don't think you should be discouraged. I know beekeeping is not that easy. About 40, 50 years ago, it was relatively easy. You would ignore your hive for 6 months and it would be still alive. But you can't expect that to happen now because of so many stressors we have, including this mite. So there is a more management required and more monitoring and um, more concentration you need in terms of beekeeping. So I'll go a basic question. Probably you might have, I'll ask you a question here. So let me talk about this population dynamics here. So this is a question for you just to think about. So have you ever thought, I said there is a reproductive phase, right? Where these mites are inside those cells. Have you given some thought? Probably you have read in some books as well. So just to think about, you don't have to answer, just keep that in mind. I'm just giving you a lot of options here. So what percentage of mites are in the capped cells here that I would call reproductive phase? And what percentage of mites are on the adult bees like this, which is called phoretic? So is it 50-50, 80-20, whatever? So just think about those. And yeah, if you want to answer in the chat, that's great as well, if you want to. But I'll come up with an answer soon. So what we did in this study is, I know you may say, people know this, why are you doing this study? It's a very tedious one. And again, it's not just for fun. Don't want my students to waste time and me wasting a lot of money doing these kind of things. Is each region is very different when in terms of population growth, right? Your bees in California may grow in a different way in May, June, July, August. And in Oregon, it rains probably until May sometimes. And our growth is different than your growth in California. So each region needs to understand their population dynamics as well. That would be helpful for your mite management and other things. So this is the reason why we wanted to understand this phoretic and reproductive mite numbers inside those cells. And it's very tedious. You'll see it's only 6 colonies, but you'll see how much work it is to really look into each cell. So I'll describe the study in a second here. So this is the uh, design for the study. So we had 6 colonies. We could have used 20 colonies as well, and you will in a minute figure out why we didn't use 20 and only 6. 6 is a good number for running statistics, and I think it's a decent number for a study. So these colonies received APOR, your amitraz, until March 22nd. And after that, until September, there was no mite treatment in these colonies. We wanted these to grow, bees to grow naturally as they would, and the mites also to grow as they would. So we didn't do any mite treatments until here. So, but we were monitoring levels throughout with alcohol wash and sticky boards as well, which are 2 methods, which I'll talk as I go. And then on September 14th, we went early in the morning, like 6 o'clock, when bees were still not yet foraging, because we wanted to capture all the adult bees that are there, right? Otherwise, I would not know how many bees are there and how many mites are there on them. So adult bees from all 6 colonies were killed in alcohol. So we have this bucket full of alcohol, and then we have this funnel. I'll show you how we did that. So basically, we killed all these 6 colonies and collected all the brood from those hives, and they came back to the lab and we analyzed for mites. So here you see a couple of students here and graduate students doing this. So we have this funnel where we shake all the bees in, and they go in and into this bucket full of alcohol. So they submerge and die, right? So we have, some will be there. You see this girl has a lot of bees on her because we were shaking. So probably I would say that more than 90% bees were captured. Maybe there were 5 to 10% max maybe that were still flying around and we couldn't get them, but 90% is a good number to focus on. Yeah, hope this video is playing well on your end.
Yeah, it's great. Thank you.
Okay. Yeah, it's a little messy, you can see. So we all went early in the morning before they're flying, and so we are looking at each frame and shaking those bees into those funnels. And then all the brood, we segregate them and they go into another box, and then they come back to the lab. And so we freeze them until we are able to look at those frames. So these are those bucket full of bees with alcohol. And then this is the one I was saying, it's very tedious. I mean, if I use 50 colonies, it would be great sample size. But again, do I have that much time? So this took us, I have like 7, 8 different projects running each year. And if I spend all the time on this project, that's not fair. So students were taking some time in between other projects. So it took about 1.5 years for us to really finish. Also, here they are meticulously looking at each cell that has a capped cell, and they're looking into the entire mite family there, and then removing those mites, counting and storing them as well. So we still have those mites stored if we want to do any future genetic analysis or any aspects that we want to look at. So we gave them some of these head magnifying glasses. So no one has sued me until now. So hopefully they didn't lose their eyesight. So they're still okay. So here you can see when they remove those pupa, you see those mites on them. So that's how we count and store those mites. So again, this is another image of a family. This is just one family inside the cell. So when you pull, you can see some of them haven't hardened yet. The exoskeleton, that they have to get to this stage to really survive and thrive. So if they're still like this, sometimes you may have seen on your sticky board some mites that are like white colored, right? So those are still immature ones. That's what you need to understand. They're not still fully, they have not sclerotized yet. The exoskeleton is not sclerotized. They're not reddish brown, but they're still a little pale, yellowish or white, whatever you want to say. So there is a male as well here. So this is another image where you can see when we pull the developing bee outside, you can see the mites on them directly inside the cells. There is one there. So again, for your curiosity, this is what we found. So the phoretic, that means the mites that were on the bees, about 44%. 56% in the brood. So just to make your life easy, you can just say 60%, 40%. But then again, this may vary. If I am doing this in July, it may vary. So this is just, so you have to understand, this is not something written hard in stone. Get me wrong here. This is in September if I'm doing in the valley here in Oregon, in Corvallis area. This is what you should expect in that time of the year. That's what the pattern is. But again, in any case, it's more than 50% for sure if it's the brood rearing season. So it may change as you get into August and September because there is less brood. So there will be more phoretic mites and less in the brood. But if you're in the peak brood rearing season, maybe in California from April on, May, June, July, you should expect something more than 50 for sure. So that's a take-home message. More than 50% mites are in the capped brood during brood rearing season. So why does this matter? Is it just a fun fact I'm saying? No, because when you are using a miticide, especially the new beekeepers in the audience, I want you to be thinking about this. If you're using like oxalic acid, maybe some of you are using it, right? So that will only take away the mites at that point of time, right? So, but then suppose oxalic acid works for 2 or 3 days. What happens after that? The mites are still in the cells, right? Every day, if your queen has been laying 2000 eggs in a day, so I would expect 2000 bees coming out every day, the new ones. And maybe half of them have mites. Maybe 1000 bees have mites in them. So you have these mites going on. So unless you are using a treatment that like amitraz, which probably is working for 42 or 56 days, whatever the recommendation is, so it slowly acts on those mites and kills. But if you're using something like a snapshot thing where it works only for a week or 2 days, then you have to be careful that you have to repeat the treatment again. Just don't think one oxalic acid treatment is enough, because that will kill only the phoretic mites at that point of time. After that, there are every day new mites coming out because they are in the cells, right? So that's what I want you to think about mite management when you are using different mite treatments that are available to you for use. This is something a little more distractive here. It's not connected to the study, but I just wanted you to see. I know many of you use alcohol wash, so there are mite monitoring methods, right? I just wanted to, because that's relevant to your beekeeping as well, is you have to monitor for mites. So sticky board is the easiest one, but it's not one of the robust ones because you're getting a big picture of it. But again, you don't know exactly what percentage of mites are in the hive, right? It depends on whether you're doing a 24-hour count or a 48 count. So it's pretty variable. So it's not a very robust way, but to just get an overall picture, yeah, we have mites. The sticky boards may serve your purpose. But if you want to really get good numbers, the alcohol wash is the best one, but next followed by your sugar shake. Sugar shake is pretty close to your alcohol wash method as well. We did some studies. I'm not gonna talk about those results today, but here I'm gonna show you, even with the alcohol wash you do in the field, shaking is very critical. If you just do very quickly, like 30 seconds or a minute, it's not enough. So we have seen, we do some alcohol wash in the field and we bring the same sample back home, back to the lab, and then we do spin on the shaker here in the lab, and you get more mites out of them. So shaking is a very critical thing when you're using alcohol wash, and I'll show you how we do in the lab, and probably Alina does at UC Davis as well, something similar. Yeah, if you have never done alcohol wash, I know you don't have to have all this fancy setup I'm going to show you here, but I'm just trying to show you that the shaking part is important, and this is how you look for mites when you use alcohol wash method. So here Sarah is, so we got some samples from beekeepers. So we do a lot of sampling for beekeepers as well. If they don't have the tools, then they send us samples, and we do in the lab. So I have a diagnostic lab too, where we look for Varroa, Nosema, and we want to look at viruses in the next couple of months as well. So you see, she's pouring all those here, right? So you have this, there is a mesh here. I don't know if you can see well, but there is a conical mesh where the bees are gonna be underneath. So now you'll see when she removes that. So it goes onto a shaker. So this is a shaker where it will shake for, I think, about 250 revolutions per minute RPM for about 2 minutes. So that's your RPM there, 250. And then, so now Claire is showing you how she does. So that's, you see, that's what I was talking about, the mesh, the conical mesh here. So the bees are all there, right? So the mites have all fallen into the jar. So she's using the sieve. So now the mites are on the sieve now because she's washing them and putting back. So no mite is left in the jar. And now she will show you how to get those mites out. So all your mites are in the sieve now, and now she is gonna put that alcohol again on top of those, cleans the sieve, and then most of the mites are now on this jar here. So you'll see them now. See all those mites, and now she's gonna use a pipette and then suck each of them, and she'll start counting at the same time. That's 1, 2, 3, 4, 5. So that's probably one of the best ways to really get an accurate number of mites in your colony. But again, as I said, don't take this seriously and try to buy a shaker and try to do those things. It's just showing how the lab techniques are done. And so we get a lot of mites. Probably 95% of the mites we can dislodge by shaking. So shaking is a critical thing. That's what the take-home message is. You need to shake your vial very well when they are in this alcohol. Okay, a little bit about the treatments. I'm not gonna go in deep. Probably you have already learned about these either at your California Master Beekeeper program or from your other mentors that you may have or read books. So Amitraz is still a synthetic one, which again, there are positives and negatives about this. APR, at least with our studies we have seen, is more effective if you use in spring because the mite levels are still low. But if you use in fall where the mite levels have already gone I mean, they have exponentially increased because it's a slow acting product. It's 3.3% amitraz, right? So it does work. I'm not saying it doesn't work, but it's more effective if you use in the spring because I work a lot with commercial beekeepers as well. And some of them have changed their strategy. They use amitraz mostly in the spring, and then they will use formic acid or other options they have in the fall. So amitraz is still a good product if you want to use. It's a synthetic one. Then I think Midaway Quick Strips is slowly gone now, so it's mostly Formic Pro. And there are two different ways to apply this. There is a 21 dose, a 21-day dose, a single dose, and then that means 2 pads. But or else you can use a single pad for 10 days. So that's what is becoming more popular because formic can be a little more toxic to your bees. If you're, especially in California, wherever you live, I would expect unless you are on the coast, maybe your temperatures are pretty high in 90s maybe or more in most of the June, July, August period. So yeah, so these are a little more temperature sensitive products, the Formic and ApiGuard. So you have to be a little more careful when you use this product because if you are using two pads or 2 strips, whatever you want to say, it could be a little toxic, and you'll see bees they will come out and they will beard outside, and sometimes the queen is not attended. So that's a major problem I see with Formic is the queens, as you know, the biology, the queen is always having that retinue of bees, right? They're always attending the bee, either feeding them or grooming and licking the pheromone. But when you use formic or so those little toxic products, there is a little bit disturbance in the hive and the bees are all gone out, or they are running here and there and they are just hiding in some places where there are less fumes. Then the queen gets ignored, right? And the queen, if it gets ignored for 2 or 3 days, that's enough for her to stop laying eggs, because queens have to be fed every probably in 3-4 times in every 5 minutes. And if she doesn't get that protein, probably there is a good chance that ovaries will shrink as well. And that's what I see from beekeepers is when they use formic, then probably they don't see any eggs for another week or so. Probably that's the reason is why the queen has not been attended by these workers for a while. So there are some consequences, but Formic is one of the best products I have seen in terms of efficacy. It does kill mites inside the cells as well. So maybe you have to find a time, or especially many commercial beekeepers now, at least in Oregon, are using only 1 pad every 10 days. So it's a two dose thing, right? So 10 days and then you use one more, which will be less toxic. The fumes will be less when compared to using 2 pads at the same time. ApiGuard is another nice product, again, temperature sensitive. So you should be careful using, look at the label and use the temperature range if it's okay when you're using it. HobGuard, we haven't done much. I did some work when the initial HobGuard came, which was a little more messy product, and now they have improvised their product. I don't think it's that messy. It's easy to apply, but again, HobGuard, I still think you need to apply probably 2 or 3 times, not just 1 dose. And oxalic is becoming more popular now because once beekeepers have used oxalic for a long time as an off-label compound getting from a hardware store, but now there is a registered product available. And I think there was a label change coming up. Maybe you guys have more knowledge than me. I haven't followed up on that. So there was a label change that was supposed to happen soon, where you can use oxalic acid even when the honey supers are on. Because the FDA has removed that limit of oxalic acid uh, in the hives. So what I'll do in the next few minutes is focus on oxalic. Even this year, we are trying to look at oxalic acid efficacy, because there are studies that have shown. So right now, the label dose is 1 gram per brood chamber. So if you have 2 boxes, then you're using 2 grams of oxalic acid dihydrate, right? If you are vaporizing it. But 1 gram, many studies in Europe and even from University of Florida, I have seen a couple of studies. They think 1 gram is not enough to get a good efficacy for mite control. So they are recommending, and they're not recommending, at least the studies I have seen, they're going at least 2 grams or even higher. So we are trying to do a similar study here in Oregon this fall. Not fall, actually, maybe by end of July, we'll be doing it. So we want to see if the dose is an issue. So maybe 2 grams is the right dose. Right now it says only 1 gram per brood chamber. So I'll give you more information, but this is a study that we looked at because if oxalic acid is being used when the brood is present, and especially with the label change, now you can use whenever you want oxalic acid. So we wanted to look at, there was concern among beekeepers that does it impact the larva, right? Probably you also have thought about it. Does oxalic acid vapors impact your larva or eggs? So this is what we did. We wanted to look at the impact of oxalic acid, but we used only 1 gram in this study. So that's Carolyn, one of my research assistants, showing you the device that we used. And you see that's oxalic acid in that. So please, if you're using oxalic acid, I know it looks like little excessive, but oxalic acid vapors are toxic. Your health comes before your bees, I would say, right? So you should agree with that. So please be careful. I know it's not a joke. I have seen some people, it's okay, the wind is blowing the other direction, I can just quickly do this. But we don't know the impacts on how it will crystallize in your lungs. And also, please wear this respirator and or something good for protection, so that you don't inhale those oxalic acid vapors. So that's what we do. And I know some beekeepers probably don't use, this is called a spacer. It's a 2-inch more space we are creating between this bottom box and the bottom board here. So I have seen some beekeepers, if they use it depends on what type of device you're using. There are several prototypes that are in the market. So if you are using something thick, then this may go, and if you didn't have a spacer, this gets very hot, right? This part. So it goes and touches your bottom of the frame, and you can create a hazard there. It may burn. So your frames get burned as well, and you may see some melting wax falling on it. If the melting wax falls on this oxalic acid, it may not even vaporize, right? So that's not a good strategy. So in our study, we use this spacer like this one, so that you can put it inside, so that it's not touching the bottom of the frame. So please try to do that if you have. But if you have a device that doesn't touch, you're fine then. But I'm just telling, if depends on what type of device you're using. So these are few master beekeepers from our program. So we do involve them in our research sometimes. If so, they get some credits for being involved in the research. So we use their expertise or service to do some of the data collection. So what they're trying to do here is, uh, so here they are looking at the cells that we had marked. So what we are looking at is we picked 2-day-old larva and 4-day-old larva. So we wanted to see if the oxalic acid has done any damage to those larvae. So we have put those marks. So these are called acetate sheets. I don't know if you see my cursor, but that's an acetate sheet. So I think this is a better image. So you see these acetate sheets, there are some red dots and there are some blue dots. So these are for different age groups. So we can remove this after we are done, and then we come back next day and then exactly place, we know we mark these pins as well. So we can exactly place this sheet again tomorrow, and then watch if that red dot, if there is a larva absent or present. So we can see if the larva is dead, then bees are pretty good, right? They'll remove it from the cell. So that's how we wanted to see if the larva was still alive or not. And we looked for eggs, larva, and later larva, I mean 2-day-old larva and a 4-day-old larva as well. So here are some preliminary results. We haven't published this yet, hopefully in the next few months. So you see percentage of eggs. So we looked at eggs as well, right? We marked some eggs too to see if there is any loss. So you can see after 24 hours, this is your formic acid treatment. And this is your oxalic, the one that looks purple on your screen. So this black one is the control. That means they didn't get any treatment here. So you can see about 35% eggs were lost after 24 hours. So it's all cumulative. Don't think this is separate data. So after 48 hours, if you're looking, about 65% eggs are gone in formic, but oxalic and your controls are pretty close to each other. They're not significantly different. So same after a week. So there is still loss. You may be surprised, why are control ones being lost as well, right? So there is an explanation. I'll see if I have time at the end to address that. But the whole point I'm trying to make is there is no difference between your oxalic and controls. That means your oxalic at 1 gram is still safe to your larva. So that was eggs, right? Sorry. So this is about eggs. What you're seeing now is about 2-day-old larva. So that's what I call them, young larva. So similar pattern, you see the formic. So formic, as I said before, formic is toxic to larva. And so that's what you should expect, some brood damage when you use formic. But that's what I mean is you have to see this balance. Is it worth killing some brood and getting rid of most of your mites? Or is it, so it depends on when you use formic. If you're using formic late in the year when your winter bees are being raised, and I'll talk about winter bees in a minute. So then that's thing you have to look for. If you're killing more winter bees and your bees don't have a chance to raise more bees for winter, then that's not the time you should use formic. You have to look for a time. Again, I know in California you live in different regions, and so your climate is very different. You have a different microclimate in each area. So see if you can find a window if you're using formic acid. So you don't want to kill too much brood late in the season, because then you will not have enough bees going into winter. So you have to do it a little early, where even if you kill some brood, your queen has still chance to lay eggs and you'll have a stronger colony by end of fall. So same pattern here, you'll see there is a larval damage. About 70% 2-day-old larvae were gone in formic, but there is no difference between oxalic and control. Similar trend with, but again, the scale is lower here. Pay attention here. It's not that bad. So it's only 25. So it looks like the older larvae are a little more resilient to formic and even the other treatments as well. So you see only about after 48 hours, about 18% loss in formic. And these are around 5 to 6%. No difference between control and oxalic. So again, take home message, oxalic acid vaporization at the recommended dose appears to be safe to the brood. But then I'm talking about only 1 gram. So this year we are gonna do 2 grams and even 4 grams as well to see if that's the case or not. So I'll go over a few important things, especially for the new beekeepers. I know some of you who are veterans here, probably I'm just preaching to the choir here, but for those new beekeepers, Varroa control is very important. So you need to continuously monitor mite levels. Continuously means not every day. I'm talking about at least once in 15 days, or if possible, once in a month during the peak season. So you need to, because mites can't be ignored. So continuously, and it could be any method. If you want sticky board, it gives you a broad sense of what's going on. But again, if you want a real sense what's going on in a hive, you need to sacrifice some bees, at least 300 bees for the alcohol wash. Or if you don't want to kill any bees, then sugar shake is fine too. You can use that method as well. So this is one thing I want to emphasize. And I know many commercial beekeepers, they don't have time, and some of them, they think it's okay. So you monitor your mite level. Suppose on July 20th, I am doing a mite treatment. So I want you to do a mite wash and see how many mites are there. And then do after the treatment, maybe formic or amitraz, whatever you use. After a month, you do again and see if it has changed anything, right? If you don't, then just assume that your product is working. Then you have lost the precious window. You are just ignoring those mites, and they have probably gone beyond your control now. So always try to monitor before and after treatment. That tells you if your product is working or not. And even a 2-week delay in applying my treatments, have seen can cause a significant damage. A good example will be, so I have some beekeepers in Oregon that have 10,000 hives. So they can't treat 10,000 hives in a day, right? They still have 15, 20 people working for them, but they can't get to all those hives in time. So we have seen even the hives that were treated on, just I'll pick some numbers. If suppose they treated some hives on July 30th, and then the others got around August 12th, I can see a big difference between those bees, how they go into winter. So even a 2-week delay can sometimes make a huge difference. And because these mites are not just feeding on the bees, right? They're transmitting viruses as well, the deforming virus and several other critical viruses, which eventually kill your bees. So yeah, be mindful of these mites and try to monitor them as much as you can. Okay, I think I'll skip that point here and see how I'm doing with time. Looks like it's already 43. So I will quickly go over nutrition. This is another area, as Wendy mentioned, honeybee health, honeybee nutrition, and pollination. So nutrition, at least last 6 years, we have been focusing on honeybee nutrition is one of the understudied areas, and it's such an important thing. I always say, nutrition is the first line of defense. If you have a lot of stressors on your bees, like same thing as us, right? If we are eating right and our immune system is better, probably you can take even Delta variant of COVID as well, right? I mean, nutrition is critical, and that's important for bees as well. It's the same immune system they have, and they have to have a better nutrition as well. So here, I'll not focus on macronutrients. You know about carbohydrates, your nectar and honey. Protein source is their pollen. But this is what we have been focusing at Oregon State for the last 5-6 years now, is micronutrients, and especially the lipids and sterols here. So I'll quickly go over a study on sterols and then go into some diutinous bees information, which is very critical for you. So pollen is their primary source of protein. As you know, bees don't get their protein from anywhere else. It has to come from the pollen. And pollen basically has crude protein, amino acids, and several other important things like lipids, minerals, vitamins, and sterols. And I'll focus on this today. And then I'll go over diutinous bees. Diutinous means the bees that are raised during late fall that will survive the winter for you. I know California has very mild winter, not as Oregon or Minnesota somewhere else. But still you have diutinous bees that are raised close to your winter time, and those are very critical. And nutrition is very important. I'm not saying nutrition is not critical now, but it's more critical when you're raising your winter bees, because if they are in good shape, then your bees will be there for spring when it's the springtime comes. So pay attention to those times. You don't want any pollen shortage in your hives when they're raising those winter bees. So these are some, again, nutritional challenges, especially for commercial beekeepers. They will deal with pollen abundance because some crops are pretty bad. They don't produce enough pollen for bees if they're going for pollination. Pollen diversity is an issue. You need a lot of diverse pollen because not all pollens are made equal, right? There are some amino acids missing, and this pollen they may get. That's why the bees have evolved and have this beautiful system where they're not just collecting one type of pollen, right? You'll see, if given a chance, they will collect 4 or 5 different types of pollen coming into the hive, because they don't know the quality. They can detect the quality of nectar. So if you have 10 petri dishes with 10% sugar, 20%, 30%, after some time, after random foraging, you'll see more bees on the highest percentage of sugar. But with pollen, they have to have some experience with it to have a preference. But if they have never been exposed to that pollen, and I'm giving them 10% protein, 20% protein, 30%, they can't differentiate the protein content. So that's a problem with the bees. That's why they collect different types of pollen, so that there is something missing and this one can be compensated with the other one. So diversity is important in case of bees as well, for in terms of pollen. And of course, quality, like we have a lot of hybrid carrot seed production here in the Central Oregon area, which is not a very nutritious crop. It doesn't produce much pollen and nectar for bees. So these are some challenges in terms of nutrition that you may encounter in your landscape. So sterols, why are they important? And what is your cholesterol? Your cholesterol, sorry, your sterol is cholesterol, right? So cholesterol, we all think it's bad, but cholesterol is not always bad. It's too much cholesterol probably clogs your arteries, but you need cholesterol for several things, for your cell membrane and all those things. And for insects, I have mentioned some things here. The sterols in insects are useful for they're components of cell membrane. They're precursors of molting hormones. These molting hormones are very critical. So the insects have to molt, right? Your day 1 old larva has to become 2-day old, 3-day old. So you learned in your literature from your master beekeeper program or somewhere else that each we call them instars, right? There is a 1-day, 2-day, 3-day, 4-day, 5-day, and then they get capped and they become pre-pupa, pupa. So for all those stages, you need these hormones. There are 2 hormones called, let me go to the next slide, ecdysone and juvenile hormone. So juvenile hormone keeps your larva in the same stage. So if the juvenile hormone levels go down, then your ecdysone goes up. And that's when you switch from 1-day to 2-day old. Then your juvenile hormone will be there. And then when it goes down, your ecdysone goes up. Then you switch to day 3. That's how it happens. So again, I'm making it very simplistic here for you, but these are the 2 major hormones that are controlling the growth of your larva or pupa or adult. So these molting hormones are very critical in insects, including our honeybees. And so if you don't have sterols, then you will not have any molting hormones. That's the bottom line. That's why you have to pay attention to sterols. And where does those sterols come from? Most pollens will have sterols, and they are different amounts. It's not all pollens, as I said, are not made equal. So they may be in different ratios or different proportions. So this is the major sterol. It's a mouthful, 24-methylene cholesterol. That's the sterol that our honeybees are looking for. And that comes from the pollen. So to just understand, so let me play this animation quickly so that you get a better understanding of sterols here. So that's your pollen. So I'm showing that in a pink dot there. So your bees come, they eat the pollen, and those pink dots are representing your sterol. So they get, so it's called endogenous, right? They're getting there into their bodies. Now they're feeding the larva. So when they're feeding the larva, they will transfer the sterol. So you see the larva also has some sterol now, the pink dots. So that becomes a normal bee and you get a healthy bee out of it. But what happens when you don't have any sterol? So again, I'm not, don't get me wrong. I'm not saying protein supplements are bad. I'm just picking an example here. So if the protein supplements don't have sterols usually, so your bee doesn't have any sterol in it because she fed on the protein. Now she's trying to feed the larva, but the larva doesn't get any sterol because the bees don't have it, right? So that's how the bees have to transfer it. So the larva will just shrivel because it doesn't have any sterol. So it doesn't molt into the next stage. So it just dries up and the bees will remove or cannibalize it. So molting hormones are very critical, and the molting hormones are directly related to the sterols that you get from your pollen. So we tested some pollens like almond, which probably you can relate. You are in the almond region. 80% of almond is grown in California, Central Valley. So we took some pollen and you see it has a decent amount of 24-methylene cholesterol. And we tested one of these protein supplements and we couldn't find anything in there. So again, we wanted to understand, again, I'm not going, we did several studies, but lack of time. So I'm just going to quickly go over this one study. And so we looked at this 24-methylene cholesterol, like how much bees need, because we still don't know how much they need. And but that's an important sterol for them. So this is a lab study. You see these bees in cages with the water and sugar. And so we gave them different concentrations of sterols, 0.1, 0.25, 0.5, 0.75. So this is total percentage of the diet. When I say 0.5%, it's 0.5% of the total protein diet that I'm giving them. So this is the work done by Priya. She's a postdoc in my lab. So this is a publication. You can look for it. If you can, then I can send you a PDF to Wendy and she can circulate. So because of lack of time, I'm not going to go into details, but it's published. You can just Google for that study and find it. So basically what we found is when we're feeding those sterols, they were selectively, they would figure out. So the consumption was higher in the higher sterol groups. So that means bees are able to perceive the sterol and they're eating that diet more. And then This is the most important part. So even though we gave them even 1% diet, but the survival was highest in the 0.5%. So you don't have to go too high in sterol either. A 0.5% is the limit that we think is an optimal concentration of sterol that the bees need. Again, these are very basic studies right now. So eventually we want to come to a place where we can provide some recommendations for these protein supplement manufacturers on how to incorporate sterols into these protein supplements. So diutinous bees a little bit, because I said I'll talk about this. So there are physiological differences, right? Those diutinous bees don't look like fat like just this in cartoon, but so basically they are same bees that you look in summer and winter. I mean in fall they look similar, but only thing is there is less larva to feed, right? And in August or September your queen has started slowing down, so there is a ratio of less larva and there is more bees, right? So the bees are, instead of storing whatever they eat in terms of protein, they are just storing everything in their bodies. But in June, July, August, when there is a lot of larvae, so the bees are not storing anything in their bodies. All those nurse bees, whatever protein they eat, they try to provide it to the larva. They're less selfish at the time. Again, I'm using the wrong term here, but just to give you an understanding of what is the difference between a summer bee and a winter bee is, in summer bee, bees are not storing anything in their bodies. But they, in fall or late fall, when they're raising those winter bees or diutinous bees, there is less larva, so they're feeding and also storing a lot in their bodies. That's why they can store and survive longer. So all this fat bodies and this vitellogenin, which is a very complex glycolipoprotein, they help them survive longer, because that's a major physiological difference, is they're storing everything in their bodies in fall and late fall. So again, this is not a good example. I'm showing you a forager versus nurse bee, but these are the fat bodies. So if you dissect a worker bee, like a nurse bee in summer, and then from the same colony, take some nurse bees in September or something late fall, you will see a lot of fat bodies in them like this. Whereas in summer, you'll not see too much fat bodies because they're not storing anything for themselves. And that's a big difference in survival. So if you have a lot of fat bodies, they will survive the winter well. And this is just an example to show you that well-fed larva, sometimes I know this is not obvious always in a colony, but as a new beekeeper, you go in and you should see your bees, your larva floating in that much of food. But if you see something like dry, you see they are like totally dry. They are just stretching out because this larva moved out because she doesn't have any food here. So this is a symptom of starvation. If you see like this, that means your colony doesn't have much pollen. So you should be looking at some protein feeding to these colonies that are showing dry larva like this. So take home message, I think I'll stop after this and take questions. I don't think we'll go into the smoke part unless you want to stay a little bit later. So make sure your bees have access to adequate pollen throughout the brood rearing season. And most protein supplements, I mean, some companies sell you 5-10% pollen added. I would prefer those rather than straight protein, which doesn't have any pollen, because most pollens have this sterol that I'm talking about, 24-methylene cholesterol. So try to, if you feed, I know some of you probably don't like feeding your bees. If you live in an area where there is abundant pollen, I don't recommend feeding. But if you think you live in places where there is a shortage of pollen, when these bees are raising those winter bees, then probably you should be feeding protein with some pollen added. And I'll show you in the last slide, like you can collect your own pollen as well if you don't want to buy from outside companies of these protein supplements. So as I said, adequate nutrition is critical during fall and late fall when winter bees are being raised. And as I said, you can also collect your own pollen. So this is the last slide I'm going to show you. So some tips for collecting pollen. So you can always look for a strong hive. Don't collect from weak hives. You don't want to deplete their pollen source. So if you have 5 hives and one looks pretty decent, strong, so sit next to the entrance and see, and you can trap whenever you want. You have to look for a time when there is abundant pollen coming into the hives. You see a lot of pollen foragers carrying pollen on their legs. That's the time you have to trap pollen. So you can use this front porch ones like these, which are easy to install, or you can do a bottom mount, something like this as well. So these ones, I would say put them late in the evening once bees are all in, because they're pretty confusing for them to orient. You'll see when you put them, they're very confused. If you go in the middle of the day and put this on, they will struggle, and some of them will fly into the neighboring hive as well. So you don't want to create that problem. So you want to, so, and then when you install it in the late evening when bees are home, or early in the morning before they forage, so you can disengage this. So this can be taped onto the top. So once you buy this, you will know how to engage this. So now it's engaged. You see the holes, the bees have to pass through them. So this is a tray that will go here and sit here. So when they go through the holes here, then they will dislodge the pollen on their legs and it will fall into this tray. So select strong colonies, put it at the right time. So first day, I would say, don't even put this, don't engage this. So just keep it up. So bees keep going in and they know how to get in. But next day, maybe in the morning, just go and engage this and they'll start coming through those cells, so through those mesh, and then they will orient themselves and how to get in as well. So that's a good strategy to do. But we have a lot of tips if you want. I know, I apologize, this is a small font I put here, but just Google for pollen collection, my last name Sagili, and JOV. This is the Journal of Visual Experiments. So we recently published, it's only like 2-3 months old publication. We have a lot of tips on how to collect your own pollen. And then once you get the pollen, and never trap more than 2 days max, maybe, because you don't want that colony to have depleted pollen sources. So you select a strong colony, only trap for 2 days, and then put in a Ziploc, and you can store in -20 freezer. And we have shown that pollen that is stored in -20 for a year, it's still good quality. It doesn't lose any protein content in there. So, and then you're not looking for tons of it, right? Maybe when you feed your protein supplement, just put a tablespoon of that, and then you should be enough in terms of sterols. Okay, this was a smoke part, so I'll stop here, and Wendy, I'll take questions.
Terrific. That was great. Thank you so much, Ramesh. That was a very interesting and engaging presentation. I know I learned a lot more about sterols, to be honest. So
Yeah.
I'm grateful. And when Priya gave her webinar to us, she was saying that you could buy. I don't even want to try to pronounce the name
20,
of this.
the
Yeah, the
24 methylene
24.
cholesterol. Yeah.
Yeah, but it was it
Oh
is
no, it's
very
very
expensive.
no, absolutely, and there is no place you can buy. We had to collaborate with a Canadian manufacturer, so they custom manufactured for us. Like, I think a gram would cost you probably $20,000. So
Yeah.
gram, literally
Yeah.
gram I'm talking. So it's pretty expensive. So that's not our goal. That, because we can't use pollens, because each pollen has a different concentration of 24 methylene cholesterol. So for experiments, you want something that is really cut and dry, right? That's the reason why we wanted to order it from a place. So we have control on how much we are feeding the bees. But yeah, the whole idea is pollen has sterol. So we have, the USDA grant that we got recently, we're trying to look at sterols in a lot of different pollens. So maybe down the line in 2 or 3 years, we have this vision of having a plant database where beekeepers can go and say, okay, the borage has this many parts per million of sterol, and then Phacelia has this much, or sunflower has this much. So that might be a good information for them to collect these pollen sources as well.
Yeah, it makes sense. Totally. There are some questions in the chat. Someone asks regarding a slide for, I believe, Dave, do you want to unmute yourself regarding your question, Dave?
Yeah, one of the earlier slides was showing a large percentage of eggs, and another one, a large percentage of larvae that were killed with Formic Pro. My question was, what are the chances of killing the queen or damaging the queen with that?
Yeah, very good question. So that's another thing we want to explore in the future. But for that study, we were only looking at larvae. But queens can be impacted. As I said before, the queens, what we have seen is those colonies that were treated with Formic, they don't have any eggs for at least 5-6 days. So that tells me that the queen is impacted. So, but we don't know if the queen physically has been damaged in terms of morphology, like has something do they have burnt legs or antenna or mouth parts? No one has really looked at that thing at this point. So yeah, in the future we want to look. A great question, but yeah, there is a risk for the queen as well. And when Formic, the mite away quick strips came initially, there were commercial beekeepers, they were complaining they had lost about 30% of their queens. So we don't know. So there is an impact for sure. I can't discount that factor. There is an impact of this, but we don't know to what extent. Can it literally kill the queen, or can it still just do some damage and leave it there?
Thank you.
Yeah, great question. And I want to acknowledge the time. It's 2 after 8. So if anybody is on here and needs to leave, please do, and know that this will be recorded. And thank you for your patience, and thank you, Ramesh, for sticking with us and answering some of the questions here.
Yeah, no problem. I'll take all questions that you have. No problem. I know, I apologize. I know I put too much information because I know I want you to get as much as possible. But your questions I can even address via email if you want to send some. Yeah.
Terrific.
Wendy,
thank you. Yeah,
Wendy-
Sandy, go ahead.
Okay, great presentation. Thank you so much. So my question has to do with feeding pollen and winter bees. So here in California, like you are also experiencing up in Oregon, we are having very hot temperatures. We're in a drought. We're in dearth already, and it's mid-July. So the queens are already slowing down, what I'm seeing. And it seems like you don't want to be raising Varroa. So do you just let them slow down, and do you not feed them until, say, September? Or what happens to the bees at this point? Because if there's not more bees being raised, they're going to decline in population, but that's also the natural part of the life cycle right now. I don't know, I'm rambling. I guess my question is, do I feed now or do I wait until September?
Yeah, no, great question. I don't think I have an excellent answer, but I'll try. Yeah, I mean, think about it as a brood break. I know the mites wise, I think you'll be in a good shape because there is a break because queen isn't laying that much. But I think I would still recommend feeding sugar because you need them to survive, because carbohydrates are also very important,
Yeah.
right? I know I didn't talk about carbohydrates because our project was more focused on pollen. But I think you probably are okay not feeding protein at this time, but protein becomes more critical. I don't know exactly where you live, but if you think that's the time when your winter bees are raised, because you need a decent population to go into winter, right? So that's the more critical part. If you have a break for a couple of weeks here, I would not really want to stimulate them. But in places like Oregon or somewhere more temperate weather, where if they lose that opportunity, then those hives are going to go in a small size, right? Then that's not ideal for Oregon. With only 4 or 5 frames of bees, they will not survive our winter. So your winters are mild, I know. And so you have to think about all those things. I know that's why I said I don't have an excellent answer, but I'm just trying to make you think about your situation. So,
Sure.
So carbohydrates are important. Please feed them sugar, because then, then they will starve and die, because they need for their metabolism every day, they need carbohydrates for sure.
Yeah, definitely feeding sugar, but
Yeah,
the protein was the question.
Yeah,
and just one more follow-up question. Because here in California, we're having such warmer winters, we're seeing the queens, they're not really going dormant. They're not having that brood break, late December. So I'm confused as to when do the winter bees actually start becoming winter bees? It seems like it would be later in the year as opposed to, September, October, or August, September. It seems like it'd be more like October, November.
Yeah. So yeah, that's another great question. So yeah, I know it's hard to really define in more tropical, subtropical climate like yours, where there is no real winter. So anything I would say, imagine that is when the temperatures are a little low, whenever it's November, December, you think you're getting into 50s or 60s. Probably that's the time. Just before that probably would be the time for your winter bees, given the biology of the bees that we understand would be. But again, yeah, you're right. If there is a continuous incoming pollen and resources are there, they will probably be like tropical climates. If you go to India or some parts of Africa as well, there is no real winter there. So then it's hard to say when that diatoneous bees are raised. But in California, I would say still when your temperatures are hovering around 55, 60s, probably just before that would be, a month before that would be your diutinous bees that are being raised.
Okay, so maybe I don't feed in late August. Maybe I push it back a little bit.
Yeah,
Okay.
if you think your queen is still laying, and that's the time when, so in Oregon, there is a window from, I tell our beekeepers, anything between August 16th until October. So the queens still lay here until October 15th or 20th. So there is a window. It's not, I can't really say. So we did some cool studies. I don't have time today. We painted bees that were released like every 2 weeks. Like we painted bees that emerge in August, then August 20th. Then so we paint them with different colors and we release them in a foster hive and see who survives by March. And so we see that the bees that are raised mostly in September, like late September until October 20th, those are the ones we find mostly still alive when I look back in September, in March next year.
Oh,
So,
that's interesting.
yeah, so you can do some cool studies. I don't know how much patience and effort it is, but you can always bring the frames and hatch like 100 bees and then give them a different color. So they don't sting you when they are 24 hours or less, because the stinger is not hardened yet. So you can literally hold and just put a yellow dot on them. And then you release them into your foster hive, then go again in September, paint some 100 bees and release them. And in March, you can go in a cooler morning and then pull a frame and ask another person to help you count how many bees, yellow bees you see, red bees, whatever you painted. And that's one way to find out what's the exact time when your winter bees are being raised.
Great. Okay. Thank you so much. And
Yeah.
again, a very, very great presentation.
Yeah. Thanks,
Great.
Sandy.
Yeah, good question, Sandy. We've got 2 more folks in the queue here. I'm going to invite Peter and then Bob to unmute. Thank you. Thanks, Pete.
Yeah, I had a couple questions back to the Formic Pro treatment. Did you do the 10-day treatment or the 14-day 2 pads?
Yeah, that's a 14-day 2 pads.
Okay. So would you expect your numbers to be, I would assume, less impactful if you did the
Yeah, absolutely. Yeah. So yeah, even our hives now, that was for the study because that we thought that's a more common recommendation, 14-day twice. So that's why we went with that. But for our hives, we still use only 1 pad every 10 days.
Yeah.
So we have found, and even several commercial beekeepers in Oregon, they find that as less disruptive to the hive, and they feel that still it works. I mean, they are still getting a decent control with 1 pad every 10 days. Yeah, twice.
But it doesn't kill under the cap, does it?
Yeah, that's a study which we haven't done, so I can't really say anything, Sandy, there. But I would still expect some control. I mean, in the brood as well. Maybe that would be a good question to answer next time we do a study, to uncap some brood and see if 1 pad also is doing the same job as 2 pads.
Yeah, you still get a pretty high concentration in that first couple of days when you put that pad on.
Yeah,
that's been my experience. Okay, and then I had another question real quick about the washing bees. I know Randy Oliver's kind of shifted away from alcohol and started using soap because he finds that works actually a little better for washing. Do you have any thoughts on that?
Yeah, no, I think I have heard his talk and seen some of those different products he has been using, from detergents to whatever. Yeah. So again, I think the percentage of alcohol, so what we use is 70% ethanol. So 70% and above, anything does a good job of exactly what probably he has done with other products. So again, if you find something cheaper and it's still working, but I haven't done any other product testing except for 70% alcohol that we use. And with that, we get a decent, probably 95% to 100% mites will come out with so again, as I said, it's a time factor as well. It's not just the product alone. If you do it and keep it for like at least 2 minutes shaking, probably you have a better release of mites when compared to doing a 1 minute shake. So shaking is, I would say, is the more priority for us than the product itself. So most of these products that he tested are, if you go lower percentage of alcohol, then yeah, getting them killed in that short period and dislodging them takes time. But if you're using 70%, I would say still you get 95 to 100% mites out.
Okay. And lastly, gig 'em.
Yeah, Aggies. Did you go to Texas A&M?
I did.
Cool, yeah.
Thanks, Pete. Bob.
Yeah, just a technical question about the freezing of the pollen. You mentioned 0 degrees can go up to a year. That's
minus 20, the freezer.
Oh, minus 20.
Not the fridge. Yeah, I said minus 20. Yeah.
I'm sorry. My freezer goes to 0. So how long's if I collect, how long is it at 0 degrees Fahrenheit?
Good question. I still think it should be pretty decent if you're, yeah, we haven't tested that part, so I can't really say what happens at 0, but I still think it should be probably a little bit different in terms of so it's all about protein denatures if you're subjecting it to sunlight, right? So if it's in the freezer at any of those cooler temperatures, I would say, again, I'm speculating because we haven't tested at the temperature. Then the data I have is at minus 20, hasn't changed anything in terms of crude protein. But even with 0, I would say there is not be much change, maybe a small percentage change here and there. Again, it's my speculation because I didn't do any data collection at that temperature.
Yeah, right. Because I just have a household chest freezer and it's in the garage, and I don't get down that low. So
yeah, that's fine. That I think you should be still fine. Yeah, I don't think there is any drastic change between that temperature and what I'm talking about. So it's more about sunlight. Yeah, if you put in the room temperature or wherever the sun is there or something, then the protein denatures faster when it's exposed to sunlight. That's a major one. And then the cooler temperatures, of course, will protect other things as well.
Thank you for your great talk.
Yeah, thanks, Bob.
Yeah, we really appreciate your time this evening with the California Master Beekeeper Program, Ramesh. Thank you. And I would love to have you back to just follow up on the research that you're doing in Oregon on the effects of wildfire.
Yeah, that's, I think I'll have more data. So yeah, that's fine. I couldn't cover today, but I feel, yeah, I have lived in Oregon about 12 years now since I moved from Texas A&M, and I never thought Oregon would be impacted by forest fires that much. And yeah, we literally didn't go out for about 2 weeks, and the air quality was 350, 500. And that's the time we collected bees. It said, okay, it's an unfortunate event, but let's collect some bees. So we collected some queens where a beekeeper was in the fire path. So he had some hives that were just close to the fire. Everything was burned. And then he had just like few hundred yards other side. They were subjected to the heat, but the colony survived. So we collected queens from those to see if the semen has changed in quality. And yeah, we did a lot of cool stuff with that. So maybe next time when I speak, I'll go over that. Yeah.
Yes, I'd be most thankful for that. And we, as a program, all of our members really appreciate, again, your generosity of time and service and insight in support of the California Master Beekeeper Program. Thank you for your presence here with us this evening.
Yeah, it's my pleasure, Wendy. Yeah.
Catching up soon.
Thank you.
All the best to you. Take good care.
Thank you. Bye.
Thank you. Great
Good night, everyone.
Good night.